Hi there. Today we are going to talk about cellular respiration. So, when we think about cellular respiration, we definitely think that it involves energy. So, you want to think about what the source of energy for cells is. Which molecule provides energy for cells? So, think back to when you had your lower level biology class. What molecule was the source of energy for the cell? And that would be the ATP molecule. So ATP stands for adenosine triphosphate, and that is the energy source that cells use at a cellular level to do the work that needs to be done. And when we talked about metabolism, we talked about how we can couple the breakdown or hydrolysis of the ATP with the reactions that need to occur in the cell. So again, ATP can break down or hydrolyze, and when it hydrolyzes, it removes the phosphate group. And as it removes that phosphate group, energy is released, and the energy that's released can be coupled with other chemical reactions. So it can provide the energy necessary for those chemical reactions to occur. Once it has hydrolyzed, then you end up with an inorganic phosphate, the ADP molecule, and then, of course, energy that you're using for things to happen in the cell. So ATP is a cyclical process. We are using the hydrolysis of ATP to do cellular work. So all kinds of things. It could be muscle contraction. It could be active transport. All of the work that needs to be done in the cell that requires energy gets its energy from the hydrolysis of the ATP molecule. After that has occurred, then we have ADP and inorganic phosphates left over. So what the cell will do is it will then restore the ATP. It puts that inorganic phosphate group back on the ADP. And that is what cellular respiration is all about. So cellular respiration is putting the ADP and the inorganic phosphate back together again. And so today we're going to talk about cellular respiration and how that process happens. So again, think back to what you know about the different organelles. Which organelle or where does cellular respiration take place? And that happens in the mitochondria. So cellular respiration occurs in the mitochondria. Later on, we're going to talk about photosynthesis. That occurs in the chloroplasts. The two of those together help to get energy for the cells. And so cellular respiration will take the energy from the organic molecules from the food that we eat, and it will convert it into ATP energy. And as a result of that, it gives off carbon dioxide and it gives off water. Both of those are then used in photosynthesis. So that's kind of a cyclical process as well. The sun's energy is used by the chloroplasts, and it will pick up the carbon dioxide and the water, and it uses those to make the organic molecules. And again, we'll talk about that a little bit later in another slide deck. So the organic molecules that we're using, that energy that's stored, that potential energy in those chemical molecules, are then used to make ATP in the process of cellular respiration. So we're breaking down organic molecules, and we are using that to generate ATP. So a catabolic pathway. So to understand cellular respiration, we kind of need to take a look at the mitochondria structure. And the structure of the mitochondria is that it has two membranes. So it's a double membrane organelle, and it has an outer membrane and it has an inner membrane that's all squiggly inside. So it's kind of like a big balloon all shoved inside of a little balloon. So it will be all wrinkled in the inside. And the reason why it's wrinkled on the inside is because the enzymes that are involved in cellular respiration, or at least the last stage of cellular respiration, are embedded in this membrane. And so having that greater surface area to volume ratio, increasing that surface area allows for more enzymes to be embedded in that membrane. The more enzymes you have, the more energy you can produce in your cells. So okay, sorry about that. So again, the more inner convoluted you have of that membrane, the more energy that cell can produce. Okay, and so it's got an outer membrane, inner membrane, and then you've got inside of the inner membrane is a place called the matrix. And then in between the inner membrane and the outer membrane is the inner membrane space. And we'll take a look at each of those as we work through cellular respiration. So cellular respiration has 3 stages, each of which contributes to the production of ATP. And so the first stage is glycolysis, and that is when the glucose is split in half. So it breaks it down into 2 3-carbon molecules, and those are called pyruvate, also pyruvic acid. So some places you'll see it as pyruvate, some places you'll see it as pyruvic acid. The second stage is the citric acid cycle, and that completes the breakdown of the glucose molecule. And then the final stage is oxidative phosphorylation, and that's where the majority of the ATP is made. Okay, so as we look through the diagrams, they'll be kind of color-coded. So glycolysis will be colored the teal color, and then the pyruvate oxidation and citric acid cycle will be color-coded salmon, and then the oxidative phosphorylation, which includes both the electron transport chain and chemosmosis, you'll see that as purple or violet. So looking first at the first stage, glycolysis, that's going to take place in the cytosol. So that means that outside of the mitochondria is where glycolysis takes place. And that is the breakdown of glucose, or the splitting of glucose in half. So glucose, as you know, is a 6-carbon molecule. It gets broken down or split in half into 2 3-carbon molecules. In the process of that occurring, ATP is made. So a little bit of ATP is made. 2 molecules of ATP net are made, but it also makes some electron-carrying molecules called NADH. And so those, think of electrons as an energy-carrying molecule. So a bundle of energy in those electrons. So that NADH is going to carry those electrons, and they're going to be used a little bit later down the line. Once the pyruvate has split in half, the glucose is split into 2 pyruvate molecules. The pyruvate is going to go into the mitochondria, where it gets broken down the rest of the way. So first of all, the pyruvate will get oxidized, and it will get converted into acetyl-CoA. And so it loses a carbon dioxide carbon dioxide comes off, and it loses a carbon molecule in that stage. Then it goes into the citric acid cycle, where it gets broken down the rest of the way. And so pyruvate oxidation and the citric acid cycle, that's the second stage of cellular respiration. And in that process, 2 ATP molecules are made. And then also a couple of electron-carrying molecules are made. So it's going to make NADH and FADH2, both of which are electron-carrying molecules. So again, bundles of energy that are going to go to the final stage, which is the oxidative phosphorylation, electron transport, and chemosmosis. So again, oxidative phosphorylation is both the electron transport chain and chemosmosis, and we'll talk about what those mean in a little bit. But as you can see in the top part of the diagram, you've got those electron carrying molecules. Those electron carrying molecules are what drive the oxidative phosphorylation process. So the most important part about glycolysis in the citric acid cycle is making those electron carriers so that oxidative phosphorylation can take place. ATP is made in all three of those steps, but the majority of it is made in that last step. The ATP that's made in glycolysis and the citric acid cycle is made in a different way, and it only makes a little bit. Those are made by substrate level phosphorylation as opposed to oxidative phosphorylation. And we'll talk about what each of those means, and we'll see how those are different. So looking first at substrate level phosphorylation, what that is there is an enzyme that will add the inorganic phosphate onto the ADP molecule, but that enzyme's doing it one at a time. And so it's not as efficient. So it has to just put one phosphate group onto the ADP and it lets go. Then it's going to pick up another one and it's going to do that. So substrate level phosphorylation is basically building the ATP molecule one at a time, or at least phosphorylating it one at a time. And so that's why in those first two stages, you're only making a little bit of ATP that way. The last stage, ATP is made very differently than that. So not substrate level phosphorylation. All right, so that's kind of an overview. Then if we take a closer look at exactly what's happening, again, glycolysis is going to break down the glucose into pyruvate. And so you're splitting that sugar molecule into two. And it actually happens in 10 steps. We're not going to get into all of those different 10 steps, but just know there's a series of reactions that take place, catabolic pathway that takes place to break it down. And that can happen without oxygen. So oxygen is not necessary in order for glycolysis to take place. And then there are two phases to glycolysis. There is the energy investment phase, and then there is the energy payoff phase. So it does take a little bit of energy to get it going, but in the end, it gives off more energy. So it uses 2 ATP, gives off 4 ATP. So we say there's a net of 2 ATP in that process. But it also makes those NADH molecules. So it's making 2 NADH, it's making 2 ATP overall, so that those can then drive, the NADH can help drive the last stage. And then once the pyruvate is created or generated in glycolysis, it will then move into the mitochondria. So it needs to go into the mitochondria to get broken down even further. And so there's going to be enzymes that will help doing that. But what will happen is the pyruvate will enter the mitochondria, and then coenzyme A will combine with the pyruvate. And in that process, one carbon is removed and acetyl-CoA is formed. So carbon dioxide is given off in that step. Then the acetyl-CoA will go into the citric acid cycle, also known as the Krebs cycle. So some diagrams you'll see it labeled as the Krebs cycle. Some diagrams you'll see it as the citric acid cycle. They're just two names for the same thing. And so one pyruvate will go through the citric acid cycle, and it will generate 1 ATP, 3 NADH, and 1 FADH. And then because you get 2 pyruvate from every glucose molecule, one glucose will actually be 2 turns of the citric acid cycle. So in the end, you'll get 2 ATP, 6 NADH, and 2 FADH for one glucose molecule. And so at the top here, you can see the oxidation of the pyruvate and the conversion of pyruvate into acetyl-CoA. So that's what happens as soon as it goes into the mitochondria. Again, carbon dioxide is given off, the NADH is made. Then the acetyl-CoA goes into the citric acid cycle, goes through a series of steps. And it's a cycle because that end molecule is then going to combine with the acetyl-CoA again. And as you can see, carbon dioxide comes off, NADH comes off, ATP comes off, FADH2 comes off, those electron carriers come off, and the ATP molecules are made. And so that's kind of the citric acid cycle, the general gist of that. And then if you look at a detailed step-by-step process, you do not need to memorize this, but just know that there's a series of steps. And then you can see at each of those stages. So step 3 is where the NADH is coming off. Step 4, more NADH is made. Step 5 is where the ATP is made. Step 6 is the FADH2. And then step 8, NADH is made. And then, of course, you can see where the carbon dioxide comes off. One comes off before it enters the acetyl-CoA, before it becomes the acetyl-CoA and goes into the cycle. You can see another carbon dioxide molecule coming off at 3 and another one at step 4. And so that is the complete breakdown of the glucose molecule. But you don't really need to get into all of those steps. Just know it's a series of steps, but it's a cycle because you're reusing those molecules again. So in step 8, that final molecule will then combine with another acetyl-CoA and it will cycle through again. Okay, so the NADH and the FADH2 that come from the breakdown of the glycolysis through, the breakdown of glucose through glycolysis in the citric acid cycle, those energy carrying molecules, those electron carrying molecules are going to be the energy that's going to drive the last step. And so part of that final step of oxidative phosphorylation, you can see that the first step is the electron transport chain. And the enzymes that are involved in the electron transport chain are embedded in the inner membrane of the mitochondria. So the citric acid cycle is going to occur in the middle of the mitochondria in the matrix. And then those electron carrying molecules are going to go to the membrane, and that's going to drive the electron transport chain. So the electron transport chain is embedded in the infoldings of the mitochondria. So all of them are in that inner lining, and that's where the electron transport chain will occur. And it's kind of like the electrons will get passed down the electron transport chain, kind of like playing hot potato. So you're passing those hot potatoes from one person to the next all the way down the electron transport chain. It's the same kind of thing, but it's the electrons that are being passed down the electron transport chain. So in the matrix in the middle of the mitochondria is where the citric acid cycle takes place. And then the inner membrane is where the electron transport chain takes place. And so this is showing you all the enzymes in that electron transport chain, all embedded in the inner membrane of the mitochondria. So here at the top, you can see the NADH and the FADH2. They are dropping off those electrons, and they're going to get passed from one to the next all the way down the electron transport chain. And then this is where the requirement for oxygen comes in. So at the very end of that electron transport chain, the electrons will end up in the oxygen. So the oxygen is going to pick up those electrons and combine with hydrogen ions to form water. So this is where you're making the water. This is where you're requiring the oxygen. Without that oxygen present, there's nowhere for the electrons to go. So we need that oxygen to pick up those electrons so the electrons can keep flowing down the electron transport chain. So the electron is the final the oxygen is the final electron acceptor in the electron transport chain. So those electrons have to go somewhere. Without the oxygen, it's kind of like a highway when they have a big accident and it shuts down the entire highway. Nothing is moving. And so if nothing is moving, then you're not going to be generating any ATP. So we need that oxygen to keep those electrons flowing. And why those electrons are important for the electron flow is because as they are moving down the electron transport chain, those enzymes are also pumping hydrogen ions into the inner membrane space. And so they're going to build up in the inner membrane space. Let's go back to this diagram. So you can see between the inner and outer membrane, there's a teeny tiny space here. And so the electron transport chain is pumping hydrogen ions into that teeny tiny space, and it's increasing the hydrogen ion concentration in that space. So they're going to want to diffuse back into the middle of the mitochondria where they're in a smaller concentration. So as the hydrogen ions are being pumped across these enzymes into that inner membrane space, it's building up that hydrogen ion concentration. And so what chemosmosis is it is using that hydrogen ion concentration gradient to drive the ATP synthase enzyme, and that synthase enzyme is generating a whole bunch of ATP. So it's kind of like a hydraulic dam. When the water flows through the hydraulic dam, it turns the turbines and it generates a whole bunch of energy. So it's kind of the same thing. The hydrogen ions will build up in the inner membrane space as a result of the electron transport chain. And then they flow through the ATP synthase enzyme. And as they flow through it, they're kind of generating that ATP. So that kind of drives that flow of hydrogen ions through that enzyme, generates the ATP molecules. And it's making a lot more than with substrate level phosphorylation. So we get a lot more ATP from this process. So this diagram kind of shows you both the electron transport chain and chemosmosis in that oxidative phosphorylation process. So here you can see the inner membrane of the mitochondria, and the top part is the inner membrane space, and then the bottom part is the middle of the mitochondria, the matrix. So all of these enzymes are embedded in the membrane, and they're passing electrons from one enzyme to the next. And as they're being passed down the electron transport chain, they are pumping hydrogen ions into the inner membrane space. And so you've got a high hydrogen ion concentration in there, and they want to flow from high concentration to low concentration. And so the only way they can get back into the matrix is if they go through the ATP synthase enzyme. And in that process, like a hydraulic dam, it is generating a whole bunch of ATP. And so that's called chemosmosis, because you are using the diffusion of those hydrogen ions through that protein, that enzyme, to generate the ATP in chemosmosis. So the electron transport chain is sending the electrons down and pumping the hydrogen ions into the inner membrane space. And then they're flowing back through the ATP synthase enzyme, and that turns the enzyme and generates the ATP. We require oxygen in this process because that is picking up those electrons in the end. It's the final electron acceptor, and it's making water because the oxygen is combining with the electrons and the hydrogen ions, and they form water in the end. So oxygen is necessary for oxidative phosphorylation to take place. Okay. So the hydrogen ion gradient is referred to as a proton motive force, which is emphasizing the capacity to do work. Okay. So if we look at what exactly is happening, cellular respiration, the energy is going from the glucose to the NADH to the electron transport chain, and through proton motive force, it gets or generates ATP. So it's making that ATP, the energy is going to that ATP. The ATP can then be used for cellular work. Okay. So we typically say that somewhere around 30 ATP molecules are made from oxidative phosphorylation. So you get 2 in glycolysis, you get 2 in the citric acid cycle, and you get somewhere around 30. But it's not exactly an exact number. So some textbooks will say 32, some will say 34, some will say 28. Just know you're making about 30 ATP molecules in that process of oxidative phosphorylation. So it's not an exact number because you're kind of generating it as it churns the turbine. And so it's not an exact number, but it's somewhere around 30. So again, you get 2 from ATP, or sorry, 2 ATP from glycolysis, 2 ATP from the citric acid cycle, and somewhere around 30 from oxidative phosphorylation. So a total somewhere between 30 to 34 ATP are being produced in cellular respiration. Okay. Then there's also a process of fermentation. So you want to think about, when does fermentation occur? Fermentation is going to be when there is no oxygen present. And fermentation is also known as anaerobic respiration. And so when cells require energy, but there's no oxygen, this still allows them to get some source of ATP energy so that they can still function. So without that oxygen, the electron transport chain is not going to work. But glycolysis itself doesn't require the oxygen. So that part of that process can still occur. So glycolysis will still be able to occur. The pyruvate just won't go into the mitochondria to get broken down even further. And there are 2 types of fermentation. There is alcohol fermentation and there is lactic acid fermentation. So if we take a look at each of these, the first one is alcohol fermentation, and some organisms will do alcohol fermentation and some organisms will do lactic acid fermentation. Alcohol fermentation we'll see in yeast and some bacteria, and that is what we use to make things like beer and wine and alcoholic beverages. We are using those organisms like yeast to make the alcohol. So what will happen, there will be, when there is no oxygen present, then glycolysis will still occur. You're making ATP molecules, 2 ATP molecules, so that the cell can still get some energy. But if there's no oxygen, the pyruvate will not go into the mitochondria. It will get broken down further. And so carbon dioxide will come off as that pyruvate is broken down. And then as a result of the breakdown of the glucose, ethanol, which is the alcohol that's in your beer and wine, that is produced in alcohol fermentation. So if there's no oxygen present, glycolysis will still take place, but the pyruvate gets broken down even further to produce alcohol. The other type of fermentation is lactic acid fermentation, and this will happen in our cells. In our muscle cells, when we work out too hard and our cells, muscle cells, are not getting enough oxygen, then lactic acid or lactate will build up in our muscles. And that's what causes our muscles to feel sore the next day, is because that lactic acid built up, and it takes some time to get rid of that built up lactic acid in our muscles. So again, what happens if there's no oxygen present in our muscles or other parts of the body, and we still need the energy requirements? The glucose will get broken down, ATP will be generated, but that pyruvate will get broken down into lactic acid. So that's the other type of fermentation. So we've got alcohol fermentation and we have lactic acid fermentation, both of which are continuing the breakdown of the glucose molecules, the pyruvate, if the pyruvate cannot go into the mitochondria. Okay, so aerobic respiration is cellular respiration because it has that air or oxygen, and anaerobic respiration is lacking in oxygen. And so the anaerobic respiration is only going to make 2 ATP, and you get those from glycolysis, while cellular respiration or aerobic respiration generates somewhere around 32 ATP. So a lot more efficient than fermentation or anaerobic respiration. So this diagram is just kind of showing you that process. If you have oxygen present, so the glycolysis is going to take place whether you have oxygen or not. But if there is oxygen present, then the pyruvate will go into the mitochondria where it gets broken down the rest of the way and cellular respiration will occur. If there's no oxygen present, then that pyruvate will go into either lactic acid fermentation or ethanol fermentation. So fermentation will occur if there is no oxygen present. So it's just showing you with or without oxygen. So aerobic respiration on the right or anaerobic respiration on the left. Okay, and then the other thing that you need to know is that we don't just eat glucose. All of the foods that we eat can get broken down and can enter into the steps of cellular respiration in different locations. So we don't just have to rely on our carbohydrates to give us energy. So this diagram is kind of showing you what will happen with the rest of the foods that we eat. So the carbohydrates will get broken down into sugars like glucose, which will then go into the stages of cellular respiration and get broken down to make energy. But we can also use the proteins from our diet. Those will get broken down and they will enter into different stages of cellular respiration, but can still be used to generate energy. And same thing with fats. Fats will get broken down and they will enter into the stages of cellular respiration in certain areas, different areas in the carbohydrates, but all of your foods, your proteins, your carbs, your fats can all be broken down and go into the stages of cellular respiration and generate energy for your cells to help make ATP. So it's not just the sugars. We just typically talk about the sugars to keep things simple, but all the foods that you eat can be used to make energy for your cells. Okay, just let me know what questions you have, and I'll do my best to answer your questions. And then you want to kind of take a minute to quiz yourself by filling in the blank diagram of cellular respiration. See if you can fill in the stuff that we just talked about. So here is that blank diagram. So take a few minutes to see if you can fill in that diagram. And then here is the answer. You can check your answer in the end. All right, that's all I have for you for today.